Thank you. Hi, everybody. Good morning. I want to say thanks to everybody for sticking around to the end, and especially thanks to the organizers. It's been a wonderful meeting, and appreciate all the work that went into pulling this off.
You can either use this.
Okay. Thank you. Thank you. So I'm going to talk about population genomics and especially reproductive success, barriers to reproductive success in Ciscoes. And this is work from Sarah Chang's PhD dissertation. She couldn't make it today, so I'm presenting some of her work. And a former PhD student named Ciscoes from my lab. All right, so start way up in the clouds. Ecologists, especially when they're feeling really introspective, they like to think about why there are so many species, so asking these basic existential kind of questions. They like to talk about community assembly. Often ecologists are curious about why there's so many individuals in a population, so how big is a population dynamics. And in fisheries, there's sort of related questions. The wording kind of changes a little bit, but we talk about stocks and fisheries. How do we identify them? Limits to population size, obviously, is of great relevance, so stock recruitment. And a lot of my training is in evolutionary biology, so I have that piece here. We also think about why there are so many species, but through a slightly different lens of natural selection as being a causal underlying production of biodiversity. And how do the species differ? So what are the actual differences in the genomes that give rise to this biodiversity? All right, so my idea with this talk, what I'm hoping to convince you of, is that for fisheries management, fisheries type questions, we can benefit from taking both an ecological and evolutionary biology approach to trying to answer those questions. So I'm advocating here an ecology plus evolutionary biology approach to fisheries biology or management. So essentially we do our ecology, run our models, get some answer, and then we separately do evolutionary biology and get some answer, and we see if we have agreement there. This is a little bit different, my reminder. This is not evolutionary ecology as a discipline. There you're explicitly looking for mechanisms on the front end that connect the two. So it's a little different approach than I think is usually proposed. All right. To try to convince you of the utility of this approach, I'm going to use Ciscoes as an example data set. So Great Lakes Ciscoes, tremendous amount of diversity. I'm sure many of you are much more familiar with all this diversity than I am. If you're curious, if you're not familiar, the Cisco monograph from the Great Lakes Fishery Commission is a great place to study. One of the key questions in Ciscoes from a fishery management perspective is why is there so much interannual variation in reproductive success? So cohort strength tends to vary wildly across years. In an ecology standpoint, there's ways of addressing this. In fisheries management in particular, a lot of you might know Hjort's 1914, this critical period hypothesis. Anybody love that? As one potential explainer of cohort strength. Here the idea basically is that if you look across the lifespan, the early life stages of a population or a stock, abundance shifts dramatically over time. And in particular, there's this time period, this critical stage at first exogenous feeding when the population often crashes. Or if it crashes, that's a strong determinant of the eventual size of that cohort. So in this particular example, because of this critical period relative to, say, some idealized norm, there's a 50-fold change in cohort strength as a result of this critical period. Of course, there's lots of other ideas about recruitment, different models. You can't read this, but it's for my own reminder. Cushing's match-mismatch, getting at the idea of whether food is available at the right times, and so on. Lots of models, but all of these models fundamentally are focused on the idea that fish generally have this type 3 life history, so across the lifespan, the percentage of survivors at any given time decreases dramatically early. So early life stages are where the magic happens in terms of determining population size. So this is a really good place to focus if you're curious about what happens to the population over time. Okay. So we're familiar with these recruitment models in fisheries. Probably less familiar to you are these evolutionary biology models. P.B. Medawar is the one I want to talk about. In 1951, gave a lecture, eventually published, on the mutational theory of aging. Any Medawar fans by any chance here in the audience? Yeah. Okay. So this is a little bit more out there, but this is a really cool thing. Medawar was talking about this unsolved problem of biology, focused sort of mostly on humans, but trying to answer this question of why aging happens. As we get older, we tend to have physiological decline, our genes don't work as well, eventually leading to senescence and ultimately death for all organisms. But the question here is really focused on what happens over the lifespan of an organism. And the central idea is that, at least in humans, most of the reproduction happens relatively early in life, and then you have this long period of life after reproduction. That's a problem from the perspective of natural selection, because if reproduction has already happened, natural selection cannot act. So what happens late in life is not subject, in some ways, to natural selection relative to what happens early in life. So this evolutionary biology model also points to the importance of early life stages, because what happens prior to reproduction is shaped much more strongly by natural selection. So while our recruitment models are focused largely on life or death, survival of individuals, this mutational theory of aging that Medawar proposed is really cool because it focuses on non-lethal impacts that may impact the ultimate success of organisms throughout their lifespan. All right, try to convince you that that's important. So what I'm proposing here is to take Hjort and these other recruitment, ecological-based recruitment models and try to bring in some evolutionary biology, sort of a new synthesis between these two different approaches, and I think there's some complementarity there. All right, most natural selection happens early in life. All right, so some data. So we tried raising Coregonus artidae Cisco eggs in my laboratory at the University of Buffalo. We were going to do a temperature experiment, and we did a temperature experiment, raising these at 4, 8, or 12 Celsius. Here are the eggs on arrival. People who look at eggs a lot might see some things here. Some of them look okay, some of them look less okay. Just after fertilization, we try to raise these over throughout their development. And we had some success, some mortality early on, as you'd expect, but also some of these seem to be developing okay as the experiment goes on. We see what looks like a normal developing embryo. But as this continues, eventually we have a complete failure in the lab, in this experiment. The high temperature individuals died quicker, as expected, than the low temperature individuals. And what seemed to happen, especially for the ones that developed correctly, is you get to this point right at hatching, and they were having difficulty hatching. So as we see these individuals, they have the characteristic curled tail, and that's sort of an interesting thing. So we've been trying to decide like what happened in this experiment. For the students in the audience, sometimes interesting things can happen on a failure, so don't worry if your experiments fail. Fortunately, we had these hobo tags, and we, for whatever reason, bought the ones that also have light as part. They record temperature, but they also record light. And in retrospect, we had too strong of lights. This isn't the actual lighting in the system, but light levels were at like 300 lux, which is too high for Ciscoes, probably. I should have known this ahead of time. We sort of eyeballed the lights in the system, but we should have actually measured it. And the reason I should have known it was a couple of years prior to this, I had worked with one of Jason Stockwell's master's students, Hannah LaChance, did this really cool study in her master's on light impacts on Ciscoes. And if you raise them under strong light conditions, they hatch early, and there's some downstream impacts. Hannah did some really cool transcriptome work and found that some of the genes that are really impacted by too much light are things like melanoma markers and other antioxidants and things that look like the fish is undergoing stress. So she actually found the genes that are impacted by this, and it shows that there's some damage occurring. Subsequently, Nikki Berry did an awesome PhD dissertation on further looking at the actual impacts, doing this experimentally, exposing fish to UV light and looking at the impacts, and they find this really impressive outcome, sad but impressive. This is the brightness here corresponds to apoptosis, or cell death, in response to too much UV light. So fish don't like being put on a tanning bed, for example. So lots of mortality here occurring. In terms of mechanisms, what actually happens if you bombard a fish with too much UV light is one of the main outcomes is DNA damage, so oxidative damage to DNA. So you know the 4 DNA bases, A, C, T, and G. One of the bases, guanine, is if there's free radicals being thrown off, this mutates guanine into some other chemical called oxoguanine, 8-oxoguanine. And this is a problem because this is an unstable base, and it's prone to mutation. It causes cancer. Ultimately, if you have a bunch of this, you can imagine reproductive failure, recruitment failure happens. All right, so that's what happens. Fortunately, your body and fish bodies have ways of dealing with this. So oxoguanine can be repaired, and the way it's repaired is through OGG1, oxoguanine glycosylase. This is the rescuer of this. Is this important? Yes. DNA damage, including this, which is the most common type, occurs at a rate of 1 million times per cell per day in your body. Do the math there. It's like trillions of times per day this occurs in your cells. So the actual efficacy of this mechanism is of critical importance. Okay. All right. Shift back to evolutionary biology. I know I'm running out of time. We did some population genomics. I'll skip over it, but we do genome sequencing. We find differences between Ciscoes, and we can look at what those differences are. And to make a long story short, I'm showing here across the genome millions of base pairs that vary. Higher on the y-axis is more differences among species. I just cherry pick. Let's just take the absolute top difference between these out of this data set and look at what this is, okay? Is the utility of evolutionary biology, I think, is we can go and pull this out. It's on chromosome 33. I'll zoom in here on this chromosome. Here's the top hit. We do natural selection scans. This part of the genome is under natural selection, according to our tests. What is it? It's OGG1, oxoguanine glycosylase. This is the rescue, the thing that rescues DNA damage from repair. So evolution, in the Cisco context, evolution is the outcome of lots and lots of cohorts over time, reproductive success over time. So this points to the long-term integrated outcome of natural selection across Cisco species is about this kind of dynamic. If you follow that. Okay. It's a complex argument, but I think we're coming at the same answer from different perspectives. Okay, so what's happening, we think in this system, there's ecological trade-offs. So in shallow water, especially that spend their time in shallow waters, especially early on in life, there's more UV light up here. There's less lipids needed for buoyancy. There's a connection there I won't talk about. But ultimately, that points to, I wrote the slide backwards, it should be high expression of OGG1 favored. And then in the deeper water species, you have the exact opposite, less UV light, less OGG1 needed. So it's this nice ecological trade-off, we think, in the system. All right. So just summarize, I think I'm out of time just about. Cisco recruitment bottlenecks, I think UV is a really good approach, and Nikki's on the right track, so I look forward to the next work that Nikki Berry does if that work is ongoing. I think we need to consider not just mortality, but also the DNA damage and non-lethal effects, and actually quantify those. Of course, lots of drivers of recruitment failure are likely and of course are occurring. Not to say this is the only cause, but it's worth looking at. Ecology plus evolution is important. I think we could do some more with recruitment models incorporating these. And I'll just stop there. Say thanks to a lot of funding sources and folks involved in this as well. Thank you. The other kind of recruitment, selfish plug for
That was fascinating. Do we have any questions for Dr. Krabbenhof?